Welcome to Chapter 6, Microbial Growth. In this chapter, we will be fulfilling Objectives 2, providing the students with knowledge necessary to comprehend the fundamentals of microbiology, mainly microbial metabolism and growth, and Objective number 3, which is provide the students with the theory behind culturing microbes, including the use of different types of media, incubation temperatures, pH, etc. In this chapter, we'll be covering requirements for growth, culture media, obtaining pure cultures, preserving bacterial cultures, and growth of bacterial cultures. Also in this chapter, we'll be looking at the information that we learned in previous chapters and actually applying them. You're probably going, thank you, finally some application, not so much theory. Under the category of requirements for growth, we'll be looking at physical requirements, chemical requirements, and biofilms. Under physical requirements, we will have an opportunity to discuss the things that we learned about in previous chapters as it applies to growing microbes in the lab. Temperature is a prime example. Organisms have an optimum temperature. This is a temperature where they grow the best at. They also have a maximum growth temperature. It's the maximum heat or temperature that they will not die at. And they have a minimum temperature. That's as cold as you can get it and still have them grow. Here's an example. I grew up in southeastern Idaho. Several of my cousins grew up in Southern California. And one of my cousins and I were attending college in Idaho at the same time. Now, when it got cold during the winter, I just put on my winter coat and my boots and I went out, and my poor Californian cousin thought she was going to die. She had a coat, but she had bought it in California. It wasn't warm enough. We had to go out and buy her another coat, and she still felt like her fingers were going to fall off, and et cetera, et cetera. She didn't make it more than one semester. So this is an example of people having an optimum, a maximum, and a minimum range, and it's even more extreme in different species of bacteria. Now, the question is, why do bacteria have different temperatures that they prefer to grow at? Well, think back to Chapter 2 and proteins and their structures. Remember, temperature is a measurement of heat. It's a measurement of energy or the movement of molecules. So if you increase the temperature above what a protein can maintain its tertiary or quaternary structure, it's going to do what? It's going to denature. It's going to come apart, and enzymes and structural proteins are going to denature, and the organism is going to die. Now, if you go colder, molecules are moving slower, they're not moving as fast, enzymes aren't working as fast, the activity of the organism slows down and it doesn't grow as fast. Now, generally speaking, if you go to the minimum or beyond the minimum, the microbe will stop growing, but it won't die. Unlike people, if you decrease the temperature for our minimum, we die. Now, originally, when we started culturing bacteria, most of them were bacteria that were associated with humans or with our environment around us. But then we started finding organisms, bacteria and archaea, in strange places. Now, this isn't a picture of a volcanic vent in the deep ocean. We call them either black smokers or white smokers, depending upon the color of the water that's coming out of them. And there are organisms that are living next to these vents that can survive pretty close to sterilization temperatures, hotter than boiling water. And if we put them at what we consider normal room temperatures, is just way too cold for them, and they will stop growing. This is beyond their minimum requirement. So we started coming up with names for these different organisms. So if we look at this chart here, this shows different organisms or different types of organisms and the temperatures that they prefer. So here we're seeing that it's their minimum, this is their optimum, and this very quickly drops off and we hit their maximum growth range. Now, this is a compilation of different species. We've averaged their preferences, and so we come up with these general groupings. Let's start at our hot end here. These would be either thermophiles or hypothermophiles. Thermo for heat or temperature, and phile for loving. So this would be a hyperthermophile. They like it hotter than about 65, and they won't grow at lower temperatures. Normal, if you want to call it that, thermophiles, organisms that we would pull out of hot springs in different areas, prefer a temperature of between 40, and some of them can go clear up to more than 70 degrees C. These are all in. Remember, we're using the metric system in this class. Now, mesophiles, these are organisms that we agree with them as to what the proper optimum temperature would be. These are organisms that like it about the same temperature that we do, although we do extend their range. So these are organisms that can survive from about 10 degrees C to up to about 50 degrees C. Now, for reference, human body temperature is 37 degrees C. Room temperature is between 22, 24, and water freezes at 0. So that gives you kind of a reference range. Now, we have our cold-loving organisms after that. These are organisms that you're going to find growing in your refrigerator out in areas where the soil freezes during the winter, in areas where the water gets cold in the winter and it's warm during the summer, organisms that you find on snowbanks. Now, there's two different groups. Now, you notice I referred to seasonal adaptions. Freezes during the winter, warm during the summer. We call these psychrotrophs. They prefer it warmer, but they can survive down to freezing. And they can continue to grow, but they don't really like it. They grow much faster at the higher temperatures. These are your psychrotrophs. Then you have your psychrophiles, those that if you get them pretty close to normal room temperature, it's just way too hot for them, their proteins start to denature, and they die. Okay, so these are your general groups of organisms based on their temperature requirements, and it's all based on how their proteins and other large molecules function at various temperatures. Let's talk about pH. Remember that pH is a scale that we use to measure the amount of free protons, hydrogen ions, that are available in a solution. Okay, now why would that be a physical requirement? Well, if you've ever been to Yellowstone, if you go to those hot springs, you'll notice that an awful lot of them also mention that they're acidic. So even if we were to take the water out, cool it down, would you still want to put your hand in it? No, because it would eat the flesh off of your hands. You would get an acid burn from it. And back in the good old days when we used to make our own soap, they used to have lye that you could buy in the stores, extremely alkaline, very, very basic, and unfortunately children would mistake the metal cans of lye for cans of sugar, and they would swallow that and it would burn their esophagus, and if it didn't kill them outright, a lot of them had to have a tracheotomy to be able to breathe around the scar tissue. So, we as humans have a pH range in which we do best. Okay, the reason for this, going back to enzymes, the active sites of enzymes often have a side chain that is either basic or acidic, and they're reacting with the opposite on their substrate. So, for example, if I have a basic side chain in my active site, then it's probably going to be interacting with an acid group on the substrate. Similar types of things. Also, remember that the side chains are involved in ionic interactions which hold proteins together. So if you change the pH beyond what the proteins can handle, you will, that's right, denature the protein and it'll lose its function. So there are organisms that need different pH requirements from us. Bacteria and such that require the same pH that we do, neutral pH, are called neutrophiles. And if they prefer an acidic environment, they're acidophiles. And if they prefer an alkaline or basic environment, and there's not very many of those, then we call them alkylophiles. Yeah, that's a hard one to say. Now let's talk about osmotic pressure, or salt or sugar solutions. Okay, normally the insides of cells are just about 0.9%. It's actually 0.85% salt solutions. And if an organism is in a solution that has the same amount of salt, which way is the water going to go? That's right, it's going to go equally in and out of the cell. It's not going to be drawn one way or the other. Now, if you put an organism into a solution that has a higher salt or sugar concentration, water wants to go toward that higher concentration. Water is pulled out of the cell, and the cell turns into a little cell raisin. Okay, we call this plasmolysis in a cell that has a cell wall, such as a bacterium. This will kill the cell. They don't put up with being turned into raisins very well. Now, let's talk about the exact opposite. What if you take a bacterium and you put it into a solution, such as distilled water, that has less salt than what it has on the inside? Water's going to want to flow in. Now, with our cells, if we take red blood cells and we put it into a beaker of distilled water, our red blood cells are going to pop. They're going to lyse because water flows in until the poor little cell swells up like a balloon and it gets bigger and bigger, and finally the cell membrane pops. Well, a cell wall prevents this. The plasma membrane swells up, hits the cell wall, and it stops. And water no longer flows in because there's already enough water pressure and it's keeping its buddies out. Sorry, the boat's full. Can't take any more. So that's why bacteria and other organisms with cell walls can swim and live in distilled water, rainwater, and fresh water and not be harmed by it. Now, what happens if you put a little hole in that cell wall? It's like putting a hole in the dam and eventually things pop. Okay, we'll be talking more about that when we talk about antibiotics. But in this chapter, you need to give organisms the salt or sugar requirements that they need. Most organisms prefer either an isotonic solution, the same as their insides, or a hypotonic salt or sugar solution below what their cell contents are. If organisms like a higher salt concentration than about 2%, we start calling it a halophile. Okay? Because most of these organisms we find in the ocean, salt water is at about 2.5 to 3%, depends upon how close it is to the mouth of a river. These are halophiles, or salt-loving organisms. There are some organisms that need salt concentrations even higher than that, and we call them hyperhalophiles. Now, some organisms prefer an isotonic solution, what we would consider an isotonic solution, but they can survive higher levels of salt. If you think about it, where on a human would you find such an organism that can survive a wide range of osmotic pressure? That's right, human skin. Your skin bacteria grow well in a solution that's 0.9% salt, but they also do well clear up to 7% salt. They have to survive when you sweat and the sweat dries. We call these organisms osmotolerant. Halophiles, as opposed to osmotolerant organisms, halophiles have to have high salt. They cannot grow in a low salt solution. Let's talk about chemical requirements. Most of the organisms that we've come in contact with are chemoheterotrophs. They've got to have organic molecules supplied to them. They need to have other molecules supplied to them in an organic form so they can make use of them. Okay, we do have some autotrophs that we grow, and they're really, really easy to grow. All they need is tap water and a few, some light, that kind of thing. It's like growing a plant. But let's talk about these chemical requirements. All organisms need carbon, whether they supply it themselves like the autotrophs do, or whether they get it from eating other organic molecules. What do they need carbon for? Okay, everything. Carbohydrates, lipids, proteins, and nucleic acids. Now, what macromolecules do organisms need nitrogen for? What do they need sulfur for? What do they need phosphorus for? Okay. Think back to chapter 2. There's 2 macromolecule types that need nitrogen. And that would be proteins. Remember, they've got an amine group, and it would be nucleic acids because they have that nitrogenous base. What macromolecules need sulfur? Okay, that would be proteins. They have some side chains have a sulfhydryl group on them. And for phosphorus, remember ATP and all of that phosphorylation going on? We need a ton of phosphorus. So we need these 4 elements in large amounts. Okay, we also need oxygen, but we're going to talk about oxygen in a little bit. Trace elements, those are things that you need just a little bit of. You need just a little bit of iron. You need just a little bit of magnesium, usually for enzymatic functions. Okay, oxygen. Okay, we're going to talk about oxygen in great detail in the next slide because different organisms need different amounts of oxygen. And then organic growth factors. Some organisms need big, complex organic molecules. Usually it's because they've been living in association with something else that provides it for them. They can't make it themselves. So, for example, mycoplasms are bacteria that don't have a cell wall, but they have a really tough steroid-enriched cell membrane. Well, they can't make their own steroids very well. So when you feed them in a lab, you have to provide a lot of cholesterol for them. They just have to have cholesterol, otherwise they won't grow. This would be considered an organic growth factor for them. Let's go back to oxygen. We think of most things needing oxygen, especially after chapter 5, when after we go through the electron transport chain, we get a whole bunch of ATPs by running NAD plus and FADH2s through the electron transport chain. So why wouldn't you want to grow in oxygen and get all of those ATPs? Well, it all depends upon what enzymes you have. Some organisms do not have electron transport chains, and oxygen can actually be toxic to them. So let's talk about the different oxygen requirements of various bacteria and how we tell. Now, these tubes, if we haven't dealt with them in lab yet, we will be. These are called reducing media. This is media that takes oxygen out of the broth and turns it into water, basically. So, if we put an organism into reducing media, and you always have some oxygen that diffuses down through because, it's being taken out here, but we've got higher concentration here. If you have organisms that are growing just at the top, they are obligate aerobes. They have to have air, specifically oxygen. They have an electron transport chain, and they cannot scale back to what they can get from just fermentation. Okay. Now, there are organisms that can do either or. Remember my example with yeast? Okay. Your intestinal bacteria also fall into this category. They are facultative anaerobes. They can grow without oxygen, but they prefer oxygen because they have an electron transport chain, and they can do even better when they have oxygen. So you're going to see growth throughout the reducing media, but you're going to see more at the top. Obligate anaerobes are those that only grow at the bottom. Up here, oxygen kills them. They either don't have an electron transport chain, or they have an electron transport chain that uses something other than oxygen as the terminal electron acceptor. Over here, we have aerotolerant anaerobes. Growth is evenly throughout. Oxygen doesn't make a difference. Usually they've got some way of detoxifying oxygen, but they don't have an electron transport chain, so they don't care. Then you have some very odd little guys that like to grow in a little bit of oxygen, okay, but too much kills them. We call these microaerophiles. Now, a famous bacterium that is famous for causing stomach ulcers is a microaerophile. When you think about it, we do have some oxygen, some air coming down your esophagus when you swallow, but not a whole lot. Okay, there's not a lot of oxygen in your stomach, but there is some. So, H. pylori is a microaerophile. On to biofilms. In the wild, most organisms do not live by themselves, just in a monospecies colony. In fact, I can think of very few instances where that would happen. But traditionally, we've done this in the lab where we try to get just a single kind of bacterium, one species of bacterium, so we can figure out what its needs are, what it looks like, and it's a whole lot easier than trying to figure out the community. But we are now moving to the point where we have enough information about the individual species of bacteria that we can start looking at how they grow in communities or biofilms. Now this is a lovely picture. This shows the structure of your average biofilm. Now if you had it just thick, there wouldn't be a flow of nutrients through, and these guys in the center would have a hard time getting any food and also having waste get out. Well, so that's why we have kind of these tree-shaped things, and you'll see over here we've got a blob that's coming off and it's drifting off to go colonize somewhere else. Now, another interesting thing about biofilms is some of these organisms we've never grown in the lab. We've grown some of them in the lab, but others we can't grow in the lab because we don't know what they need. They're evolved to grow with their buddies, and their buddies protect them from bad influences. Say, for example, we have different species of bacteria in here, and the organisms on the outside are aerophiles, but the organisms on the inside are obligate anaerobes. These organisms are going to make use of the oxygen, and they're going to keep it away from their buddies on the inside. Meanwhile, the buddies on the inside, maybe they're providing something that these guys on the outside need. Maybe a complex organic molecule that the guys on the outside can't make. Okay, so they all live happily together in a community, and well, sometimes it's not so happy. Your skin bacteria engage in chemical warfare. They produce antibacterial agents that try to kill off each other. But such is life in the wild. One last thing about biofilms before we go on. Part of the reason why we've been interested in biofilms is they're very hard to treat with antibiotics. And we're used to having biofilms growing on the outside of us, on your skin, on your intestinal lining. You didn't think that was outside, but it really is. It's not in contact with your blood. But when they get in to your body where they shouldn't be, they can also form biofilms, and it's very hard for antibiotics to get through these biofilms. And so we've had a very strong interest in biofilms and how to break them up when they get where they shouldn't be. Well, I thought I could do this chapter all in one go, but I've talked too much. I have too many good stories. Anyway, I'm going to stop here. We will continue this chapter in part 2, in which we're going to talk about actually growing the organisms in the lab, and also how we keep track of how many they are and whether they're growing a lot or little.